Okay. Okay, welcome everyone for today's session. Before we begin, I would like to respectfully acknowledge that we're located on Treaty 6 territory, a traditional gathering place for diverse Indigenous peoples, including the Cree, Blackfoot, Métis, Nakota, the Suero, Iroquois, Dene, Ojibwe, Saulteaux, Anishinaabe, Inuit, and many others whose histories, languages, and cultures continue to enrich our vibrant community. So welcome everyone for today's grand round. My name is Gopi and I'll be chairing the session today. It is my great pleasure to introduce Dr. David Lillicrap, who is a professor in the Department of Pathology and Molecular Medicine at Queen's University, who will be giving his grand rounds lecture today. His medical education was completed at Imperial College Medical School in London, and he began his postdoctorate training in medicine here at the University of Alberta, so he has some connection here. He was the recipient of the Senior Canada Research Chair in Molecular Hemostasis until 2022, and in 2013 he was elected to the Fellowship of the Royal Society of Canada. Dr. Lillicrap is a past member of the World Federation of Hemophilia Medical Advisory Board and chair of the WFH's Research Committee. He is a past chairman of the International Society on Thrombus and Hemostasis Scientific and Standardization Committee and a past member of the Council of the ISTH. Between 2008 and 2014, Dr. Lillicrap served as an associate editor of Blood and has recently completed a 5-year term as co-editor-in-chief of the journal Thrombus and Hemostasis. His research program, which he'll talk about today, focuses on the molecular science of factor VIII and von Willebrand factor. Thank you very much, Dr. Lillicrap. We look forward to it.
Thank you very much indeed for that kind introduction. I want to thank Professor Giroudi for inviting me to come back to Edmonton. I haven't been here for a long time, but it's a real pleasure to be here this afternoon, and I'm going to be learning about what one of your PhD students has discovered in the last 2 or 3 years. Good morning, everyone online. Thanks for joining me this morning. I'm going to talk to you about a journey this morning to do with hemophilia gene therapy. So let me get started here. I'm going to try to leave about 10 minutes or so for questions. So here are my disclosures. Okay. This has nothing to do with hemophilia, at least if it does, I'm not sure what it would be. But in Kingston this Monday, we had this exciting experience of a total solar eclipse. And this is one of the pictures taken during the 2.5 minutes of totality. If any of you have seen a total eclipse, this was incredible, actually. It was really, really astonishing. And so during this time, you can actually take off your protective glasses and look at the sun with the corona that you see here. And you can see some solar flares as well in various parts of this picture. So here's a flare in this corner. Anyway, it suddenly became really cold. And then this happened. It was a remarkable experience. Anyway, nothing to do with hemophilia. So as Gopi just mentioned, our research group has an interest in 2 proteins, von Willebrand factor and factor VIII. We're interested in the biology and pathobiology of VWF. We're interested in the immunology of factor VIII. But this morning, I'm going to talk just about the translational aspects, that is, gene therapies for hemophilia A. So the first few slides are just going to be a gentle introduction to this disease. Many of you know all about this, of course, but with a gentle intro. These conditions then, the hemophilias A and B, are relatively simple monogenic X-linked inherited bleeding disorders. The genes for these conditions were cloned in the early 80s, factor IX, 1983, and factor VIII, 1984. And you can see that there are about 4,000 individuals in the country who have hemophilia. So these are rare inherited diseases. About a third of these individuals have severe disease, and I'll show you the consequences of that in a moment. And the reason that there are many more hemophilia A versus B is that the mutational target The factor VIII gene is about 4 or 5 times bigger than factor IX. So this explains the predominance of hemophilia A versus B. And I'm spend most of my time talking about hemophilia A this morning. So the bleeding that occurs in this condition is predominantly musculoskeletal. This is a severe hemophilia A child from Africa, 6-year-old, and you can see that his knees are very deformed. He's had recurrent spontaneous knee joint bleeding, resulting in what you see here. I mean, this still happens, unfortunately, in developing countries because of lack of access to therapy. There are infrequent incidences of other spontaneous bleeds into muscles, sometimes into internal body cavities, and rarely, fortunately, intracranial bleeding can occur. But this is the type of thing we see in our older patients with hemophilia in developed countries like Canada. So here's someone who's 48, had multiple bleeds into ankles, knees, and elbows. And you can see that he's now got muscular atrophy. He's actually got joint deformities. And this is the hemophilic arthropathy, which we try to prevent now with preventative prophylactic therapy. And really, the best way of actually producing prophylactic therapy would be through gene therapy, as I'll describe later in this talk. So I'm going to briefly describe some of the other aspects of hemophilia therapy before I get on to gene therapy. This is the evolution since the 60s to current day. Initially, we treated hemophilia with plasma-derived concentrates, something called cryoprecipitate. If you freeze plasma and then thaw it at 4 degrees, a precipitate falls out, and that contains factor VIII and von Willebrand factor. And so if you resuspend that, you can give it as cryo, and that actually treats hemophilia very well. And then plasma-derived concentrates of factor VIII were used up until the cloning of the genes in the mid-80s. And then, so from that point until the early 2000s, we were using recombinant concentrates. And since then, there's been a tsunami of developments, which I'll describe a little bit about in the next 3 or 4 slides. So nucleic acid-based therapies, antibody-mediated therapies, and then bioengineered recombinant concentrates. So this is the landscape in 2024, at least in a country like Canada. So we still use factor VIII concentrate a lot, either standard half-life material that has a half-life of 12 hours, and then some extended bioengineered recombinant concentrates, which have very extended half-lives, so up to now 3 or 4 times the standard half-life of factor VIII. The most recent addition of this is a compound called EFA, which is a recombinant factor VIII joined to the factor VIII stabilizing part of von Willebrand factor, the D' D3 region of VWF. And so that allows half-lives of over 40 hours. And this means that the individuals, the patients who are prophylaxing, can give themselves an injection once a week, an intravenous injection once a week, as opposed to 3 times a week with the standard half-life concentrates. I'm going to say a few more words about these in just a moment. So there are now several bispecific antibodies, Hemlibra, Mim8, and NXT007, which partially mimic factor VIII cofactor activity. We're now also beginning to use rebalancing therapies, that is inhibiting some of the anticoagulant mechanisms, antithrombin, tissue factor pathway inhibitor, and protein C, and then ultimately gene therapy. Now, for time purposes, I'm only going to go back and discuss a little bit more about these bispecific antibodies, because I think, as you'll see, that they are truly transformative therapies in the treatment of this condition. So this represents the biochemical focus of hemophilia. Here's a plasma membrane. Usually we think activated platelets. And on that is the formation of a complex called the intrinsic 10As complex. It's called that because in this complex, factor 10 is converted to 10A, thus 10As. The enzyme that does that is activated factor 9, 9A, and it does this on a surface, a phospholipid surface, and it requires a cofactor, and that cofactor is activated factor 8. So factor 8 is not an enzyme, it's basically a scaffold protein that allows the juxtaposition of the enzyme with the substrate to produce this reaction. If you remove factor 8, so this is the complex in hemophilia A, and it doesn't form because the scaffold protein is missing. And so this now reduces the catalytic efficiency of this reaction massively and makes it biologically useless. And this is why hemophiliacs bleed. So you're missing the cofactor activity. And so now about 18 years ago, scientists in Japan at Chugai found a bispecific antibody represented here, which binds through one arm to the enzyme, one arm to the substrate, and partially mimics 8A cofactor activity. So we believe the factor 8 equivalence of this is somewhere in the region of 10 to 20%. It isn't, you never will get 100%, but it's decent for preventative therapy. This can be given subcutaneously, not intravenously, every 2 to 4 weeks. It works in factor 8 patients who have factor 8 antibodies. Now you can see around the world, there is massive numbers of patients who are on this. And indeed, in this rare disease, this is now one of the best-selling drugs in the world as of 2022. It's made by Roche, by the way. So if you want to get some of the action here, Roche is the place to. So this is a very positively transformative therapy for hemophilia A. It doesn't work, of course, for hemophilia B because you need the enzyme. Okay. So now I'm going to talk for the rest of this morning about gene therapy. The genes were cloned, as I said, in 83 and 84. The first gene therapy trial was carried out in China in 1993. It didn't work, but fortunately, it didn't cause any problems. And then between 1995 and 2010, there were a number of small phase 1/2 studies using different vectors. But the major advance occurred in 2011 when a group in London and at St. Jude in the US gave an AAV8 vector expressing factor 9. And that therapy for the first time worked long term. And in fact, there are some patients now out now to about 12 or 13 years who continue to express factor 9 at therapeutically relevant levels, around about 5%, but they're not bleeding. So this was the breakthrough in 2011. Now, there are different ways of considering gene therapy for hemophilia. I'm not going to say anything much about non-viral therapy. They're still being worked upon. Certainly, lipid nanoparticles as a delivery method has some potential future. We're not at the point for mutation repair yet, although, of course, CRISPR-Cas9 is being investigated as a possibility. Cell-based therapies, maybe at some point in the future. But currently, everything that's being done is using viral gene transfer. So using the power, the evolutionary power of viruses to deliver nucleic acid to produce a therapeutic benefit. Now, the work that I'm going to describe to you has a basis on the use of these animals. So this is one of our recent litters of hemophilia dogs. These dogs have spontaneously occurring severe hemophilia A. They were originally derived from animals at the Mayo Clinic, and they've now been in Kingston for about 40 years. They're one of 2 colonies in the world. The other is at Chapel Hill in North Carolina. And we treat their bleeding. They bleed about 5 times per year and require treatment with recombinant canine factor 8. So we treat them with state-of-the-art recombinant pathogen-free canine 8. Interestingly, about a quarter of them develop antibodies to their treatment. And currently we have 45 dogs, and half of them have received gene therapy with canine factor VIII. So this is a major area of interest for us to try to understand how this treatment works and understand things about the complications. So when I returned to Queens after a postdoc, we set about cloning the gene in 1998. 4 years later, we described the mutation that produced this. When I read this title, I'm not even sure what we were describing. If you read this, it's actually very complicated. And honestly, I think we didn't know exactly what this was, but we thought it was very similar to the recurrent intron 22 inversion mutation, which occurs in hemophilia, human hemophilia. And indeed, 18 months ago, with the use of long-read sequencing, which has only been available really for the last 4 or 5 years, we now know that the dog mutation looks very, very similar to the mutation that causes 45% of severe hemophilia A in humans. And we were able to do that by long-read analysis. So here's what we did back in 2001. We used an adenoviral vector, not AAV, but adenovirus. We injected this at this dose with an albumin regulatory element in 2 dogs. And you can see that within 2 days, we achieved 20-fold therapeutic levels, 20-fold physiological levels of factor VIII. Now I've never done an experiment like this ever before or since. This was astonishing. We rarely believe this, that these dogs were so responsive. But you can see that within 7 to 10 days, they lost the factor VIII levels. And the dogs, by the way, were completely fine throughout this. There were no systematic responses. But you can see their ALT, their liver enzyme levels, show this biphasic elevation. So within 2 days, they were very high, probably because of the extremely efficient transduction of hepatocytes. And within 2 weeks, these levels are hepatitis levels. So there was significant hepatotoxicity after the delivery of this vector. And in addition, both of the dogs developed high titer anti-factor VIII antibodies. We measure those in something called a Bethesda assay. This is over 1,000 Bethesda units, which is extremely high. And dog B also developed antibodies as well. So this was an interesting experiment, but there's no way, clearly no way, that you would consider going forward with this as a potential way of doing this in humans. And we know from the studies that Jim Wilson did, many of you recall that there was a teenager, Jesse Gelsinger, who died from adenoviral gene therapy back in 1999 because of a systemic sepsis problem. So adenovirus induces a massive innate immune response. It's very pro-inflammatory, as I've just shown you in our dogs. So other delivery systems, lentiviral systems are being considered. They're still undergoing preclinical development. But currently, all of the therapies are using adeno-associated virus, AAV. So I've already told you the first successful therapy occurred in 2011 using AAV. This is an electron micrograph showing multiple AAV particles. They have about a 20 nanometer diameter. And here for comparison is an adenoviral particle, which is about 100 nanometers in size. So AAV is the current standard of therapy. And if you look at gene therapy for other conditions, including things like SMA now, AAV is being used for a variety of therapies in the clinic. So AAV, the wild type virus, is a non-pathogenic parvovirus. So many of us in the audience here today would have been infected with AAV, but you don't know it because it doesn't produce obvious symptomatic problems. There are round about 15 different serotypes with different tissue tropisms, as I'll explain in just a moment. And when you deliver things with AAV vectors, the majority of the vector exists outside of the host chromosomes. And I'll get back to this at the end of my talk this morning. So here's what you do when you take the wild type virus and you convert it into a vector. So at the top here, you see a viral genome. It's a single-stranded DNA genome of about 4.7 kilobases. There are inverted terminal repeat elements at each end of this genome, and there are two coding regions, rep and cap, within the viral genome. These are responsible for capsid production, and then the rep genes are involved in replication and integration into the capsid. When you convert this into a vector, you remove these two genes. You insert a therapeutic transgene, in our case, either factor 8 or 9. You provide a regulatory sequence, a promoter enhancer sequence, and then you maintain the ITRs, which are required for encapsulation. So in fact, this is not anything like the virus. There's only about 10% of the viral genome left. You now insert that into a protein capsid that I'll show you in just one moment. So here's the vector genome. One of the issues actually about AAV is that it has a limited packaging capacity. Any cDNAs, any things that you want to express from a transgene that's bigger than about 4 kb will not fit into AAV. That's not a problem for factor 9 because the cDNA is about 1.5 kb. But for factor 8, it means that you have to remove a significant part of the coding sequence, which fortunately is not required for a cofactor function. So this is the packaging capacity is a limitation. This then gets packaged into a capsid. You deliver this by peripheral vein infusion. And then depending upon the capsid that you use, illustrated by these different colors here, you deliver to different organs. And we want to deliver to the liver. So you'll see that the studies I'm going to show you are predominantly using actually AAV5, which is a hepatotropic capsid. Some of the vector will spill over into other tissues. So when you deliver this by intravenous infusion, it gets to many other tissues, not the germline. We know that the germline is actually resistant to AAV transduction, but we provide a liver-specific regulatory sequence to limit expression to hepatocytes. So here's the results of one of our studies published in December of 2022 in Blood about long-term follow-up of liver-directed AAV gene therapy in dog factor 8. This was the first dog to receive the therapy. This is a small cohort, just 8 dogs, all with severe hemophilia A. And we followed them up to actually beyond 12 years. So these dogs were kept until they were euthanized because they were getting old age issues actually at the time. They received the therapy through single portal vein infusions. Pre-delivery, they had no factor 8. Post-delivery, over more than 12 years, their factor 8 levels remained at about 10%. And you can see that their bleeding, which went from 4.3 to essentially nothing for a decade period. So this told us that with this delivery system, achieving low levels of factor 8, essentially mild hemophilia levels, prevented spontaneous bleeding for an extended period of time. And I think that gave companies, I think, sort of the courage to move forward with some of the clinical events that I'm going to show you in the subsequent slides. So the biggest study in factor 8 gene therapy that's been carried out so far is this Gene 8R8 study carried out by BioMarin. And this is the cohort of 132 individuals. So it's a big study. At the time, it was the largest global phase 3 study for any gene therapy product. And this was started in 2021. So these individuals, they all had severe hemophilia A. They received an AAV5 vector. This is the vector dose, which is pretty big, actually. And you see here the factor VIII levels at 1 year, at 52 weeks. And I'll just sort of summarize what you see here. Here are the factor VIII levels, the activity levels, going from nothing up to 400%. And you see that the peak levels are reached at about 6 months. There is very, very big variability. About 5% of the patients did not respond. The rest had some level of response. And you see at 1 year, the mean level was 41%, the median about 23%. So, and you begin to see that from 6 months onwards, there's a bit of a fall off, and I'll show you more about this in a moment. So predictability of the response here is a problem. We don't understand this. So we still don't really understand whether you're going to be here or whether you're going to be here. And obviously that's a problem for patients. Now, so if a patient's asking, what is my factor level going to be? You can see over here, there's 24% of patients who had levels below 5%. Some of them, 77% of this group, remain bleed-free. When they got below 3%, only about a third of those patients did not bleed. So this patient group, yeah, they did okay, but it wasn't wonderful. But the remaining 75% had levels between 5 and over 40%. So this is certainly not ideal, but the significant majority of patients had good responses and showed evidence of continued bleed-free existence. In the hemophilia community now, people like to talk about hemophilia-free mind because they would not need to receive any infusions during this time if the therapy worked. So at 1 year, the levels were 41.9 and the median at 22.9. You see at year 3, these levels fell further down to 18 and 8%. But their annualized bleed rates, ABR is the annualized bleed rates, continue to be very, very low. So despite the fact that the levels fell over the first 3 years, these levels, which are basically mild, non-severe hemophilia A levels, continue to be preventative against bleeding. So what happens subsequently? Well, this is a cohort, again, of individuals who received the same vector dose, just 7 individuals. So this was the original phase 1/2 study, who are now out at 7 years. And you can see here in this kind of unusual plot. So here's the weeks going out to 7 years. And here are the factor VIII levels on the vertical axis, 0 up to 200%. You see that, again, there was a very good response in year 1, and then the levels fall. There are multiple time points that were assessed over this time. So all of these different individual dots are the 7 patients sampled repeatedly over this period of time. And you can see now at week 8 of week 364, the mean and median levels are around about 20%. So there has been a progressive fall, but it looks as though once you get below 20%, the levels may be persistent in these 7 individuals who showed a therapeutic benefit initially. And we simply don't know. This is the longest of any experience. And remember, the dogs went out to beyond 10 years at low levels, but they showed no evidence of loss of expression at an extended period of time. In those 7 individuals, here are their ABRs. So at baseline, they were experiencing over 20 bleeds per year. Again, red is median, blue is mean. And then after therapy, their bleed rates, annualized bleed rates, were very, very low, despite at some points their levels being below 20%, which our mild hemophilia patients tend not to bleed unless they experience trauma or undergo surgeries. So this figures actually with levels around about 20% that these individuals do not bleed. And this is their annual infusion rates. So they were treating themselves either mainly on demand or sometimes relatively ineffective prophylaxis. But you see that they were not needing to infuse. I think there was one individual who had some bleeds around a procedure here. So there was a bit of an increase here. So the message from that is that despite the fact that the levels in these small number of patients, just 7 individuals, were falling, particularly in the first 3 years, the clinical response looks fairly encouraging out to year 7. Now, this is the one factor 9 study that I wanted to spend a little bit of time on, because it's very clear now that factor 9 gene therapy is different than factor 8. You can see here in this phase 3 study using AAV5, so it's the same increase in the activity as measured here. So this is a study of 50 individuals, 50 severe hemophilia B patients. And you can see that by about week 6, the levels are somewhere in the region of 30%. And they continue in a plateaued fashion out to 18 months. And these patients are now beyond, I think, 2 or 3 years. And the levels have remained very stable at about 30 to 40%. And the variability also is less than we saw with factor 8. We believe that this is probably because we're making cells, hepatocytes make a protein, factor 9, which they make natively. This is not an unusual protein for these cells to see. Hepatocytes do not make factor 8 regularly. Factor 8 is made natively in unusual subsets of endothelial cells, and that currently is not the target for gene therapy. So we think the hepatocytes are freaked out by making factor 8 initially, and they don't like it. And so they lose expression until it drops to below a tolerable level of about 20%. Okay, so in the last 10 or 15 minutes, I want to just talk a little bit about what we don't really know about gene therapy. So here's a picture of the life cycle. So you deliver a vector into the circulation. It gets picked up by receptors on cells on hepatocytes, enters an endosome. Eventually, there's endosomal release. It enters into the nucleus. Once it's in the nucleus, the capsid, the protein capsid, is uncoated and proteolyzed by the proteasome and actually presented on the cell surface. There's a synthesis of a second strand, because this is a single-stranded genome. And then the vector persists either as an episomal copy or integrated into the chromosomes. You get transgene expression into message, protein expression, and then secretion. So many of these steps, these 9 steps, we don't know much about, to be honest. There's a fairly big black box about some of these elements, and I'll return to this at the very end of my talk. So let's briefly discuss eligibility, reliability, the variability, durability, and safety of hemophilia gene therapy. So the first thing is that there are a number of patient populations that are excluded currently. If we took blood from everyone in this room, we would expect that somewhere in the region of 30 to 50% of you have antibodies to AAV. You've been exposed to the virus. You have humoral immunity and probably cell-based immunity as well to the virus. So you cannot be treated with this vector because the vector will be neutralized before it gets to the hepatocytes. We deliver the treatment into hepatocytes. So if you deliver this to a three-year-old, as the liver is growing, as the liver gets bigger, it will spit out the episomal copies. And so you get a dilutional effect. At least that's what we think, although we have actually dog results now which counter that. But nevertheless, at the moment, you have to be 18 or above to receive this as a human patient. There are no trials ongoing yet for inhibitor patients, although this is starting. And then if you failed or you lose expression after prior therapy, if you're one of the small number of patients who don't respond, you can't be retreated because your immune response is so big that any further reinfusion will be futile. Okay, how reliable is the therapy? Well, once an effective vector dose has been identified in the early phase 1/2 studies, evidence of therapeutically relevant levels occurs in over 85% of cases. I mean, the levels may be low, but very few patients have no response. So talking to individual patients, you could say that you would be very unlucky not to get some response after the treatment is delivered. Now, I've already highlighted the variability on the factor VIII study that I showed you initially. So there is very significant variability, which is unpredictable and really, I think, a major issue. And it's predominantly with the factor VIII trials. With factor IX, as I've illustrated, we can say to factor IX patients that if you get a response, which is going to happen in the significant majority of individuals, your levels will remain fairly stable once you're out to about 6 weeks. So why is this? And I've already hinted at this. There is no doubt that I think patients are going to be looking at factor IX gene therapy in a much more positive way currently than factor VIII gene therapy. And part of that is the issue about the hepatocyte choice that I've already alluded to. We know from cell-based studies that if you produce a lot of factor VIII in cells that normally do not produce it, you induce cellular stress. And in particular, you induce an unfolded protein response, which ultimately, if it's not rescued, produces apoptosis of the cells that are trying to produce factor VIII. So we need to find ways of producing some tolerance to cells that can make factor VIII under conditions such as gene therapy. Hepatocytes don't make von Willebrand factor, and factor VIII is the carrier protein in the circulation. And so one of the thoughts that we've had is actually try to deliver part of von Willebrand factor to chaperone factor VIII so that it doesn't misbehave in cells. We think of factor VIII actually as a prima donna protein. It really goes around causing trouble in very small amounts. And it has this chaperone, a big protein, von Willebrand factor, which makes sure it behaves better in the circulation. So there's an inherent challenging nature of factor VIII. In my lab, if people get asked to work on factor VIII, they don't like it. They want to work on this protein, which is a little bit easier. Yeah. How long does the treatment last? So I've told you that in large animal models, it lasts out to beyond 10 years. So both the Chapel Hill group and us have seen this for both factor VIII and IX. And in the human studies in adults, so patients aged 18 and above, the factor IX studies are out beyond a decade, and I've shown you the 7-year data for factor VIII. So I think you could say that 5% levels for 5 years is kind of a reasonable guesstimate. But if you're talking to factor IX patients, I think you could even be more optimistic. For factor VIII, I'm not really sure yet. Okay, so I'm going to finish the talk now with some discussion about this issue. So this, as you see, was actually accepted by Blood just a few weeks ago, March of 2024, and it represents the results actually of 15 years. This was not a PhD study. This would have driven a student crazy, actually. At the end of the study with the 8 dogs, we were able to do extensive autopsies and have been investigating that tissue now for the last 3 or 4 years. So safety. So the safety issues with gene therapy is short term, and there are infrequent acute transient reactions to the infusion in small numbers of individuals. So you infuse this material over about an hour intravenously, and occasionally the patients have sort of idiosyncratic reactions. They don't last very long, and usually you can complete the infusion. The biggest problem is this medium-term problem, which is liver toxicity with increases in ALT, alanine aminotransferase, which usually occurs about 2 to 4 months after the infusion. Over the last 3 or 4 years, I must have been on, I don't know, 100 Zoom calls trying to work out why this is happening. We don't really know. It occurs more with factor 8 versus factor 9. You can see 3/4 of the factor 8 patients get this problem, about 1/4 of factor 9 patients. And there is some response to corticosteroids. Probably, honestly, this is probably a cellular stress issue. Initially, it was thought to be a cell-mediated immune response, and there was some evidence from ELISpot assays that this was occurring. But increasingly, we believe this is more complicated than an adaptive immune response. It may be a combination of immune responses and cellular metabolic responses. And then the final issue is that, of course, of potential genotoxicity, and that's what I'm going to finish on. So beyond 7 years, we don't really know, and there have been very few human biopsies done on livers, about 10 so far, to begin to look at this. So when you infuse this material, it gets into the liver, it transduces predominantly hepatocytes, and it can exist either as non-integrated small concatenameric episomes, or it can get integrated into the host chromosomes. For a long time, the dirty secret of AAV therapy was, oh, it's fine, it doesn't integrate. That was a lie, and people knew that, but they didn't discuss it. We now know that, and it has to be discussed with patients. So is there a long-term genotoxic risk? So when you give AAV vector therapy, you give huge amounts of vector particles. So 10 to the 15 vector particles. That's a lot of vector. The patients, they don't have any systematic responses. I mean, occasionally there are these idiosyncratic infusion things, but otherwise it's not an issue. And a very, very small fraction of the vector is integrated. This is the number, but this number, when you look at this, is still a lot. That's the issue here. So at least 10 million integration events occur per liver, simply because the vector particle numbers are so large. These vectors have no machinery to make them integrate. These are not retroviral particles. So they integrate into the host chromosomes through passive events. As we all in this room, our genomes are dynamic. They are undergoing recombination events. They're undergoing breaks in the DNA, which is repaired. And at those events, some of these vector particles enter into the chromosomes. The vector integration, as I'll show you in a moment, is not entirely random, but it occurs all through the genomes. However, in the dogs from Queen's University and Chapel Hill, despite following those animals for beyond a decade now, there's been no evidence of any problems with histopathological evidence for liver tumors. There have been 5 cases now in hemophilia patients who've had cancers But in every instance, when the cancer's been biopsied, there's been no genomic evidence that the vector integration has played any role. So these are patients who've developed, in one case, a B-ALL. There was one liver cancer. That individual had hepatitis B, hepatitis C, and also had non-fatty alcoholic liver disease. So there have been other events which have driven the oncogenic outcomes in those individuals. Now, in the dogs that we euthanized at 10 to 12 years, we subjected them to genomic analyses. And here you see the 8 dogs. We're looking at now 2 liver biopsies in each one of those dogs. So this dog, LE1, had 2 liver biopsies. VC had 2 liver biopsies. And the blue bars represent non-integrated episomal vector-vector sequences. So you can see very easily in all of these animals, over 90% of the persistent vector sequences beyond a decade were episomes, which actually in itself is kind of remarkable. So over 10 years, there must have been some hepatocyte turnover in that time, but we still found the predominant vector form was episomal vectors. So either those are non-replicating hepatocytes, or there's some way that the episomes are traveling with the chromosomes during cell division. This is a hemophilic dog karyotype. So dogs have 38 chromosomes, and here's chromosome X. And each one of these blue bars on the small amount of liver biopsies we took shows you an integration event. They're everywhere. I mean, you can see that there are huge numbers of integration events. And you can see on chromosome 14, chromosome 28, and over here near the factor VIII gene on the X chromosome, there were increased numbers of integrations. We understand this because we think some of these were sort of failed homologous integration events close to the factor VIII gene. We still actually don't really understand why these occurred. The chromatin was open. Certainly was more accessible for integration, and that certainly played a role, but it seems to be unusually predominant in these 2 places. So the summary of these data is that episomal forms are the predominant long-term form of factor VIII AAV vectors. The integration events occurred with about 1 integration per 1,000 cells. This is less actually than you would see with lentiviral integrations, and it occurred in all animals with some common sites of integration that I've just described to you. All integrated vector sequences were rearranged and or truncated. This is important because we believe that the long-term expression is not coming from the integrated events because they look as though they're just not capable. They're structurally rearranged. We think long-term expression is coming from the episomes. The integration sites were predominantly in intergenic regions, so they haven't actually integrated into genes. And there was no evidence of adenomas or carcinomas at postmortem. So the first gene therapy approvals have occurred for factor VIII in August of 2022. That material is called Roctavian. It was approved originally in Europe and now in the US. And factor IX, the first approval by the FDA was back in 2022. And you can see the cost of these therapies. They're theoretically one-time treatments. And if you do economic analyses, after about 5 or 6 years, this would be cost saving because you're no longer giving concentrate infusions. But the one-time upfront therapies costs are a lot. In Canada, factor VIII gene therapy has not been approved yet, but factor IX therapy has been approved in October of last year with this therapy, Hemgenix, made by CSL. And in January of this year, the Pfizer product, Becvez, was approved. And so this will, we've got to find out ways whether we can pay for this in Canada, but this theoretically would be available for patients sometime perhaps in the next 1 or 2 years. So to conclude, phase 3 studies, of which there'll be some more, require a couple of years to mature for efficacy evaluation. Factor 9 studies honestly hint at long-term benefits out to beyond a decade. And I think factor 8 studies require further evaluation. There are immune obstacles in gene therapy with pre-existing antibodies which prevent you from ever receiving this therapy, and we can't redose these patients. The biological knowledge of gene delivery and expression processes is still very limited, I would say. There's still a big black box about what happens inside the cells, but none of these considerations has inhibited the licensing of hemophilia gene therapy products. And I think there are expectations that small numbers of patients will choose to undergo gene therapy in the next couple of years, particularly for factor 9, where the results are looking actually pretty promising. So with that, I've finished and happy to take any questions.
Okay, thank you, Dr. Lillycrop. So we will take questions from the audience, but I'll also look online. Yes, go ahead.
So is there any correlation between the actual mutation the patient carried and their response? Like I know sorry. Yeah, so is there any correlation between the mutation they had? So in factor 8, or were they all inversion patients versus truncating variants and their response?
Yeah, so Sherry, it's a good question. The genotype of all these patients is known. So there's severe hemophilia A's, so 45% of them will have the inversion 22 mutation. No one has done that correlation, actually. It's an interesting thought. So the variability, could it somehow play a role? I don't know. I mean, but the information is available and someone should do the analysis. I mean, there are 132 patients there, so it's not a small number. Yeah, good question.
Okay, we have one online. So increased factor 8 levels could shift hemostasis from a hemorrhagic to a thrombotic phenotype. Have any of the trials assessed the hypocoagulability in patients who experienced an overcorrection of the factor 8 levels?
Yeah.
Any thrombotic events?
Yeah. So thank you for the question. For sure, any coagulation protein in excess produces a hypercoagulable prothrombotic state. So far, the increased levels of factor 8 have been fairly transient, and there have been no thrombotic complications, and people haven't worked up the hypercoagulability that much. In factor 9, in the factor 9 gene therapy studies, there has been one patient who had a factor 9 level for a period of time of over 300%, and that patient experienced a thrombotic event. The patient was then actually anticoagulated for a while, and everything worked out, and the factor 9 levels fell. So that question is extremely relevant, and so unfortunately we have no way of predicting at the moment what level you're going to end up with. But certainly hypercoagulability is a theoretical, and in one patient, a real concern, because this did happen.
Okay.
First of all, thank you very much for this really exciting and excellent presentation, David. My question is, considering how much 1 milligram plays a role, following up on Cheryl's question, was there any study on the level of BWF in patients, whether that plays into the variability of response to factor 8?
Yeah, so certainly that's a really important question, and that analysis has been done. So they've looked at the levels of the factor 8 after the gene therapy, and there is not a correlation with the patient's existing VWF levels, which of course can be variable. I mean, some of these patients they should all have normal VWF levels, but they could be 50% or 200%. And there's no correlation with the outcomes that we're aware of so far. But it's an important point. I really think that somehow this idea that we're trying to make this factor 8 in the wrong cell is a concern, but it's not easy to get around that.
My question is, do gene therapy patients develop inhibitors?
Yes. Yeah, thank you. So the question is about whether any of these patients are they at risk of developing inhibitors. So the first thing is that the patients who've been enrolled so far on these studies are all adult patients who've been previously pre-exposed to factor VIII concentrates extensively. So they've been kind of screened out of not developing inhibitors. In fact, if you have a history of an inhibitor so far, you're not eligible. There is an early phase clinical study beginning now to look at gene therapy in inhibitor patients. I think there are about 5 patients enrolled. The evidence, so none of the patients have developed inhibitors on these studies, and there is preclinical evidence that gene therapy has the potential to immune tolerize patients. Because if you think about it, immune tolerance is we give repeated infusions of concentrate. Well, gene therapy is doing that intrinsically, and there's a constant supply. And so both in mice and in dogs, we have experience with the group actually in the States of producing tolerance in dogs who have pre-existing inhibitors through gene therapy. So I think that the gene therapy, particularly from the liver, which is known to be a sort of an immune modulatory organ, has significant potential to tolerize against this antigen and not produce new antibodies.
So the question about your the variability in terms of the expression. In your preclinical studies, is there any evidence to suggest that in the dogs, for example, where there was more integration, they have less expression versus those that set epichromosomally? So I guess to summarize, is potentially those patients that have the low expression, is it because it integrates more into their genome versus those that have higher levels where it sits epichromosome?
Yeah, so thanks for the question. I think the first thing to say is that the genomic studies that we carried out they took about 3 years. They are very complicated and costly. We actually couldn't have done this on a CHR grant, that's for sure. So there's a lot of things that need to be done to answer your question correctly. One thing I would say, though, is that we spent a lot of time on the liver samples we looked at to see what the structures were of the integrated copies. And we failed to see any integrated copies that were intact. So both with short read sequencing and then ultimately with long read sequencing, where you should be able to capture structural changes, we always saw that the integrated copies were truncated, rearranged, or partially deleted. So we believe fairly strongly, although this is still contentious actually, that the episomes are where the long-term expression is coming from. And we haven't quantified that in the different dogs. I guess we could do it. I'm not sure how we would do it actually, but it's an interesting
I just wonder if over time, as it integrates more in, you're now generating less, because as you said, it integrates, it basically destroys it in a sense.
Yes,
you know, so that you're losing the amount that are epichromosomally there actually expressing it. So that's what
I mean, I think it's a good illustration that the basic science of gene therapy still has a long way to go. I mean, essentially, in a way, it's almost surprising. It's taken 35 years to get it into the clinic, but even now, you could argue that for factor VIII, I don't know.
Or is there a way to inhibit its integration to have it just only sit epichromosomally?
That's a great point.
Okay, we have no more questions online. Do we have any final questions in the audience?
Thank you for turning out this morning, by the way.
If not, then thank you so much, Dr.
You're welcome.
for an amazing talk.
Thanks very much.